going to talk about early safety signal detections of drugs from public internet sources. Next slide, please. So I'd like to start with the problem statement. It is estimated that more than 2 million adverse drugs reactions are reported annually through various systems, and it causes more than 100,000 deaths annually. ADRs are fourth leading causes of death after pulmonary disease, diabetes, pneumonia, and automobile accidents. Currently, as per my understanding, reporting of ADR events happens to the traditional reporting systems like clinical trials, post-marketing studies, and some of the voluntary reporting that are done by the patients and the physicians. And naturally, it takes months to years to receive this information and process that information. But in the age of social media like Facebook, Twitter, those vast amount of information are currently being reported online, and very few of them are getting collected and analyzed so far. So there exists a huge opportunity to collect those data, run them through the machine learning and AI algorithm, and present the meaningful information that are extracted to the decision makers like FDA and other pharmaceutical industries to make real-time or near real-time decisions. So here, along with our project, we'll propose an end-to-end solution involving all the stakeholders like IT and subject matter experts to create a robust, comprehensive infrastructure where you can early detect those events and take appropriate actions as necessary. So for the last 2.5 months, we are building a prototype of the solution that we are going to present here today. My colleague Marty will go over data and different project phases and best practices and some of the challenges that we learned about the project. Myself will go over the modeling part, and HA will go over the dashboard that we have produced for tracking those events and reporting those events. With that, I will hand it over to Marty.
Thanks, Sanjib. Once again, my name is Marty Jolly. First, we want to express our appreciation for everyone's efforts during these extraordinary times. The PHUSE FDA leadership, our internal leadership, the previous presenters, and all who have contributed. Let's start by looking at the data, as it might provide some context for the rest of these slides. Here are two examples of sentences we worked with. The color coding highlights the mentions of drugs, reactions, and indications found. You may notice the punctuation and numerical data was removed. Social media posts vary from platform to platform. Twitter tweets are short and to the point, versus Reddit posts. Examples like these are much longer and more like storytelling. And modeling approaches may vary with the different platforms. Next slide, please. Now I'd like to dive into our process and share what we've learned about applying deep learning techniques to the challenge of drug safety surveillance. The primary goal of our work was to develop a pipeline ingesting raw social media posts and outputting potential adverse drug events found in those posts. Early in January of this year, we began our work. Our team sat down and split out the work into 4 phases: acquisition of the data, annotation, labeling of the data, modeling and prediction, and dashboarding. In the next 3 slides, I'd like to outline our approach, highlighting our workflow, challenges we encountered, and best practices identified to address those challenges. Here are the phases that we talked about are simply overlaid on our workflow chart. Our acquisition, annotation, modeling, and dashboard. After spending time to understand the problem space better, and we're still doing that, we began reading the literature and exploring the potential social media data sources. Twitter and Reddit social media platforms were identified as our starting point. We extracted a limited number of Twitter posts from the Twitter streaming API, but our focus was on the health-related Reddit subreddit channels. The Google BigQuery Reddit archives were used to acquire a year's worth of posts across 3 subreddit channels. The channels were named Medical, Ask Docs, and Opiates Recovery. These downloaded posts were post-processed and loaded into our annotation tool, where we also loaded FDA product lists and known drug side effect lists to pre-annotate the post. A side note here is to point out that all data processing was accomplished using the open source Python programming language and Pandas data manipulation library. At this point, a team of 6 annotators manually labeled posts where the drug names and associated drug reactions were found. After this annotation work was completed, the information was extracted from the tool and post-processed to format as required for input as training data into our model. Several models were trained and evaluated. The best performing modeling techniques were used to predict whether previously unseen posts contained potential adverse drug reactions. The output from that prediction was post-processed in order to format the data into a reporting format required for loading into a dashboard, which we developed. Next slide, please. Shown here are some of the key areas where we identified and began implementing best practices that would take the process to the next level of maturity. Number 1, we saw that leveraging existing data management tools to catalog our data, modeling the data in order to create the metadata necessary for an automated pipeline was important to ensure speed of operation, reliability, ability to manage, and transparency. This is especially important when there are multiple data sources. Number 2, we found it critical to protect all parties when processing potentially sensitive personal data. Procedures for ensuring rights to use from the data provider and consent to use from the content creator are important. This became a big issue in our project as our legal team identified serious risks related to using the Google BigQuery Reddit archives. It is at this point we pivoted to synthetically generated posts for this project. Number 3, we absolutely see the opportunity for using this technology for many different applications and different public internet data sources and non-public data sources as well. Number 4, building annotation teams with the appropriate levels of skill and additionally transferring knowledge from previously annotated sources is important. Quality labeled data is the first step to quality modeling results. Number 5, once we pivoted to synthetic data, we found that generating training data synthetically addresses multiple issues. We found it could boost our model's performance and provide training data in cases where there was a lack of such data or a limited amount of such data. Number 6, it's important to continually improve our models, especially as the underlying technologies are currently advancing quickly. Finally, number 7, we found that consolidating drug names and reactions into standardized name categories is critical for reporting and integration into legacy systems. Drug names and reactions can be expressed in many ways in social media posts, and it's very helpful to map them to standards. Next slide, please. And quickly here, we see a possible IT architecture and set of tools to support an enterprise-level solution utilizing the latest cloud-based compute resources and open software stack. This graphic illustrates how we have implemented similar solutions. This slide highlights an end-to-end solution which covers all the requirements from IT infrastructure to storing of the data, data registry, metadata, training of the model, implementing the model using services, using APIs to provide access to the model output for UIs such as a dashboard. Of course, any real-world implementation would be based upon the specific needs of the enterprise. Next slide, please. Switching gears, this is a screenshot of the annotation tool used by our team. The tool allows for marking mentions, relations, and co-references. We see here where an annotator marked mentions of indications in yellow, drugs in gray, reactions in red. Also, we can see relationships between drugs and reactions are marked with the arrows and the drug_ADR tag. Each annotator was given a set of posts to label. There was an overlap where posts were duplicated across annotators. Once the team finished annotation, there was a reconciliation process to address the cases where annotators labeled the same post differently. At this point, we had what is called the ground truth. The label data was extracted from the tool and post-processed to format the data in the form required as input to the model. At this point, I'll hand it back to Sanjib, who will go into the modeling details.
Thanks, Marty. So we applied a technique called named entity recognition, which is very popular in the natural language processing area. We started with a traditional machine learning model in the named entity recognition space called conditional random field, or CRF. Then we went to some of the deep learning methods. The first one we tried is called bidirectional LSDM model. Then we moved to LSDM with combination of CRF. And then final one that we applied is a state-of-the-art deep learning model called ELMO. Next slide, please. So for any kind of NLP machine learning project, we need to go with the formatting of the data and then cleaning of the data. So we pre-processed NLP statement and we use techniques like removing stop words, etc. Then we tokenize the sentences, and then we tagged the words for labeling purposes because we used mostly supervised training method. We also used semi-supervised training method, but labeling had to be done. So for the labeling, you can see on that picture, if a drug word contain only one word, then we started with the B-drug. So this is a common technique called BIO technique. If it is more than one word, then it will get, the first one will get B-drug, then second one will get I-drug. Similarly, for the reactions, you can see the back pain. So back will get B-reaction, and then pain will get I-reaction. Then we also added part of speech for the label. Then the records were fed into the model, and we started doing training and testing. We used the standard 80-20 split between train and test data. Next slide, please. The first one, as I said, that we started with the CRF technique. So CRF uses a conditional probability distribution. So basically, it is looking into the sentences, and it's trying to determine the presence of drug, indication, and reactions. So you can see that we got a very good result right away. Applying this technique, our precision score was about 0.9, and F1 score was 0.88. But one problem we were facing, so we started with Twitter sentences, and those were smaller sentences. But when we were trying with longer sentences, then we are having issues with the predictions. So that's when we thought about applying some deep learning models. So next slide, please. So then we moved to the LSDM CRF method. What it changes from the earlier CRF method, normal CRF, is the scoring functions. So earlier we were applying scoring function is the log linear scoring functions. But in this case, we moved to nonlinear neural network method, LSTM. So that improved our accuracy quite a bit. So after that, we were getting accuracy, validation accuracy in the range of like 0.97. Next slide, please. Then we went for the state-of-the-art ELMO method. So the beauty of the ELMO method, it works on the pre-trained word embeddings, and also it takes the whole input sentence of the functions. We also applied the bidirectional LSTM method with that, and got a very good accuracy here as well. Next slide, please. So this is kind of the final slide, which shows the comparison of different models and what we got accuracy-wise. Our ELMO model was the best model in terms of accuracy. So with the sentences that we had for our training and testing, we got about 0.98 accuracy with the sentences. And this is an example of what model was producing as output. It was able to detect mentions of drugs and reactions. With that, I will pass it over to Eche, who will go over some of the graphs and charts of the dashboard.
Thank you, Sanjib. So as the final step, we pulled the results from the model Sanjib described in detail, and we uploaded the data into our dashboard platform, which is IBM Cognos Insight. We chose Cognos because it is readily available to us, but this type of design can be done using any similar open source dashboard tool. So this is how it looks like, and from the model, we retrieved the reactions for each specific drug. And for this demo, we selected 11 popular drug names. So if you look at the two tables on the top of the slide, we can see the number of reactions for each drug, and then the list of reactions for each drug listed separately. And on the right side of the screen, you'll see the monthly trends, and there is a separate series for each drug. So this way we can locate any spikes and any possible abnormalities in the trends just by looking at the graph. Next slide, please. And if you look closer, we can see how all those charts are connected. For example, we can see number of reactions for cetirizine, the distribution of those reactions in detail, and the monthly trend. Next slide, please. And the colorful heat map allows the user to visually locate drugs with higher and lower number of reactions, and as the user scrolls over the drug name, number of reactions will just pop up. Thank you. Next slide, please. So finally, I want to bring your attention to what makes a dashboard so user-friendly and functional. On the left side, there is the Explorer pane, which will allow the user to make any number of selections, such as drug names, reactions, and the date, which can be quarter, month, or we can even go to a specific date. And the whole screen will change to reflect those choices. And as the data evolves, we can add more fields, such as the geocodes or drug categories or reaction categories. And this is the end of our presentation.